Bismillahirrahmanirrahim. Once again, welcome to my new lecture. So today today our presentation topic is a review on clinical pharmacokinetic and dynamics. So today we will discuss about the different clinical pharmacokinetics and dynamic in the term of half-life. These are the learning objectives. So in this lecture we will explain the basic concepts, measurements and significance of half-life. To explain the concept, measurement and significance of steady state concentration, to explain the concept, measurement and significance of clearance and kinetics of drug eliminations. So now pharmacokinetics. So as we know that pharmacokinetic, the drug molecules interact with the target site to produce effects. The drug must be absorbed into the bloodstream and then carried to the target site. As in previous lecture, we have already discussed the pharmacokinetic profile, that what the body does with the drug. So it means that in pharmacokinetic we will study about the absorption of drug, metabolisms, distribution and elimination of drugs. So here in this case, as we know that the drug molecule, they can be interact with the target sites to produce effects, and the drug must be absorbed into the bloodstream and then carried to the target site. So pharmacokinetic is the study of drug absorption, distributions within body and the drug elimination over time. Now what is half-life of drug? So half-life of drug can be defined as half-life, it is the time which is taken for a drug concentration to fall to 1/2 of its original value. And in other words, the elimination rate constant is the friction of drug is in the body which is removed for unit time. And simple words, we can say that half-life, this is the time which is required for a drug to achieve the peak plasma concentration to be reduced half 50% of its original concentrations. It means that in a particular period of time, the concentrations of the drug will be reduced up to some extent. Let's suppose if a drug, if 100 milligram drugs, the time which is required for if a drug which is 100 milligram, so it means that in 100 milligram, the time which is required for that 100 milligram drug, it will be reduced 50% half of its original concentration. So it means that that concentration will be reduced with the passage of time in a particular period of times, and then it will be reduced to half. As this definition is clear, plasma half-life T1/2. So it can be written mathematically, it can be written as T1/2 of the drug. Generally it is measured by the times, or you can say to decline the plasma concentrations of a drug 50% from the peak plasma concentration ppc by T maximum. So I will discuss about the peak plasma concentrations in detail. For example, plasma half-life T1/2 of drugs. So actually this is the time to degrade the concentrations of drug from 100 to 50 and 2 hours. So 2 hours, this is the time which is required for this drug, to achieve the peak plasma concentration that must be reduced 50% half of its original concentration. If it is 200, so it must be reduced to 50. 100. If it is 300, then it must be reduced 150. And how much time? So this is the time. So the time which is required for a drug to achieve the peak plasma concentration to be reduced 50% half of its original concentration. Now look at here, time to decline the concentration from 100 to 50 and 2 hours. So it means that the half-life of this drug is 2 hours. So this point is clear that in a period of 2 hours the concentration of this drug the concentration of this drug will be reduced to 50. So look at this is the graphical representations of the plasma half-life of the drug. So look at here. So it means this is what this is the times. So this time is required, the time which is required for a drug to achieve the peak plasma concentration. So this is what it means, that the concentrations of the drug will be reduced with the passage of time. Look at here. The plasma half-life of the drug, generally a drug will be completely eliminated after 6 half-lives. Look at here, there are 6 steps, and first ones, after one half-life, the concentration of the drug must be reduced to 50%. Let's suppose if a drug concentration is, let's suppose a drug which is 100 milligram, 200 milligram or 300 milligram, but the concentration of that drug must be reduced half after first half-life, while in after second half-life, so it must be reduced, and second half-life it must be reduced and it will be 25%, while in third case after the third half-lives the concentration of the drug will be reduced 12.5%, while in fourth half-lives the concentration of the drug will be reduced 6.25%. And fifth, so after fifth half-lives the concentration of the drug will be reduced to 3.125%, while in 6 half-lives the concentration of the drug will be reduced 1.56%. It means, so from one half-life, so when we are going from bottom to top, so it means that the concentration of the drug will be reduced. Look at here, in first half-life the concentration, what was the concentration? The concentration of the drug was reduced 50%, but in the final stage the concentration was reduced 1.56%. So it means this is very less. Furthermore, plasma half-life T1/2 of a drug is generally, it declines to 6.25%, will usually be far below the therapeutic thresholds. It means, look at here, it means that after 4 half-lives generally it declines to 6.25, which is already mentioned here. After 4 half-lives, it means, so they are talking about this half-lives. So generally a decline to 6.25% will usually be far below the therapeutic threshold. So for this reason, in order to find out this reason, it is usually said that the drugs no longer have a pharmacological effect 4 half-lives after the last dose. It means in these conditions the drugs will no longer have pharmacological effects, it will lose its pharmacological effects when the drug crosses the stage after 4 half-lives. It means it is very necessary that the concentration of the drug will be reduced up to 50%, not to this range, not in the range of after 4 halves. Furthermore, I would like to say why half-life is important. This is a question, why half-life is important. So half-life is a major determinant of half-life. It shows the duration of action after a single dose whenever we are going to administer a drug. After a single dose, the duration to find out or to determine the duration of action. That's why half-life of drug is important. To find out, to determine the duration of action. The second one is the time which is required to reach the steady state concentration. Now what is steady state concentration? Steady state concentration or steady state, that is the states when the rate of administration of drug is equal to the rate of eliminations. It means that how much quantity of drug you are going to administer, so it must be equal to, it must be that quantity of a drug must be eliminated from the drug with that momentums. So the first one is duration of action, the second one is the time which is required for a drug to achieve the peak plasma concentrations, and that is the peak plasma concentration, and with the rate of influx is equal to the rate of efflux. Mean the rate of administration is equal to the rate of eliminations. The third one is dosing frequency. Let's suppose we are using a different type of medications, OD, BD, TDS, single dose, 2 times a day, 3 times a day. You can say this is called dosing frequency. So through with the help of half-life, so you can, these are the major determinant, through you can easily, why we are using different type of medication in multiple doses. Now come toward the importance of half-life. What is the importance of half-life? So the first importance of half-life is estimation of the dosing schedules. So it defines the time interval between doses. It is very important. Let's suppose if you are using a drug for a period of, let's suppose if you are using a drug in divided dose. For example, you are using a drug BD twice a day. So it means there is 12-hour gaps present between these two doses. Let's suppose if you are using a medications or drugs in divided dose TDS 3 times a day. So it means that the interval between these 3 doses, so it become 8 hours. It means that you should take each regimes after 8 hours. If a single dose is required, so it means that you can use a single dose in 24 hours. So that's why estimation of dosing schedule, it is very important. It defines the time interval between the doses, and it is very important in the design of infusion system. So this is also very important, especially when you are going to designs of infusion systems. The second one is estimation of time to drug eliminations, that and how much time this drug will be eliminated. So with the help of half-life, you can easily determine that the drug can be eliminated from the body. And my next lecture, I will explain about the different formulas which are used to determine how we can find out or how we can determine the half-life of different drugs. So it gives idea to estimate the time to the total drug eliminations. This is the second point. So it gives idea how to estimate the time to the total drug elimination. Generally, most drugs will be eliminated approximately 6 half-lives. Now look at here. This is called plasma half-life T1/2 of some drug. These are some drugs, and with the half-life they are already mentioned here. So the half-life of benzyl penicillin is 30 minutes, while the half-life of amoxicillin is 1 hour, while the half-life of paracetamol is 2 hours, while the half-life of atenolol is 7 hours and diazepam is 40 hours. So what does it mean? What does it mean 30 minutes or 1 hour or 2 hours? Let's suppose we have atenormines, atenolols, which is used in the treatment of hypertension for hypertensive patient, and this is antihypertensive drug. For example, tenormin, this is the drug which is available in a market by a dose of, let's suppose, 50 milligram. So it means that the concentration of 50 milligram, the concentration of 50 milligram, it will be reduced 50% half of its original concentration and this period. So it means that in 7 hours the concentration of the drug will be reduced from 50 to 25%. Let's suppose a drug, for example, paracetamol. Paracetamol, it is available in 500 milligram. So it means that in 2 hours, you can say in 2 hours the concentrations of the drug will be reduced 50% half of its original concentration. And these are the drugs. Benzyl penicillin, so it has the plasma half-life is 30 minutes, while amoxicillin is 1 hour, paracetamol is 2 hours, and atenolol is 7 hours. So benzyl penicillin, so its half-life is less than amoxicillin, and then paracetamol, and then atenolol and diazepam. In next lecture we will discuss about the elimination half-life of drug, that how the drug can be eliminated from the body.